Welcome back to Edutube. Science is simple. In this video, we're going to explore microscopy. That's just a fancy word for using microscopes to look at really tiny things. Let's start with two important terms: object and image. The object is the actual thing you're looking at under the microscope. When you place some onion cells on a glass slide, that's your object. Now, when you look through the microscope, you'll see a bigger version of those cells. That's the image. Next up is magnification. There's also a simple formula for magnification, which is image size over object size. This tells us how many times bigger the image is compared to the actual object. For example, if you look through the microscope and the image looks 100 times bigger than the real object, the magnification is 100 times. Now let's talk about resolution. Resolution is the minimum distance between 2 points that can still be seen as separate points when viewed under a microscope. The smaller the distance that can be distinguished, the higher the resolution. In simpler terms, it tells us how clear or detailed an image is. Imagine you're looking at two pictures of onion cells. Both images are magnified the same amount, so the cells look the same size. But one image shows cells more clearly, and you can see individual cell walls and nuclei more sharply. Even though both images has same magnification, the image on the right has better resolution, and it shows more detail. Now, let's begin by talking about light microscopes. If you're using a microscope in school, this is most likely the kind you have. So, what is a light microscope? As the name suggests, it uses light to help us see very small things we can't see with just our eyes, like cells. There's a light source at the bottom of the microscope. Sometimes it's a little lamp, and other times it's a mirror that reflects light from the room. That light focuses right onto the specimen. The specimen is placed on a glass slide, and the slide sits on a flat platform called the stage. A light microscope uses two lenses to magnify the specimen. The objective lens is close to the specimen, and the eyepiece lens is the lens you look through at the top. Because it uses two lenses to magnify, it's called a compound microscope. On this microscope, the objective lenses are attached to a part called the revolving nosepiece. You can spin it to choose between three lenses: the scanning lens, which magnifies 4 times, the low-power lens, which magnifies 10 times, and the high-power lens, which magnifies 40 times. The eyepiece lens also magnifies usually by 10 times. So if you're using the scanning lens and looking through the eyepiece, the total magnification is 40 times. You're looking at something 40 times bigger than it really is. Now that we know how the microscope makes things look bigger, let's learn how to bring the image into focus. That means making it clear and sharp. Take a look at the two knobs on the side of the microscope. The bigger knob is called the coarse adjustment knob. It moves the stage up and down in large movements and is used to quickly bring the specimen into view. Then there's the smaller knob, right next to it. This is the fine adjustment knob. It moves the stage up and down very slightly and helps you make the image really sharp and clear. Some microscopes also have stage knobs. These let you move the stage side to side or forward and backward, so you can explore different parts of the slide without touching it. But not every microscope has these. Sometimes, you'll need to move the slide gently with your fingers. Now, before you use the microscope, let's talk about handling it safely. The microscope has two main support parts: the base, which is the bottom part, and the arm, which connects the base to the top of the microscope. When you carry the microscope, always use two hands. One hand should hold the arm, and the other should support the base. And remember, if your microscope has a plug and needs electricity, make sure the area around it is dry. Now, let's talk about a different kind of microscope, the electron microscope. Unlike the light microscopes we use in classrooms, electron microscopes are much larger, more expensive, and a lot more complex to use. Electron microscopes are used by trained scientists in advanced science laboratories. But what makes them so powerful? Well, instead of using light, electron microscopes use electrons to create images. Electrons have an incredibly tiny wavelength, about 0.1 nanometers. That's much smaller than the wavelength of light, which means electron microscopes can pick up much finer details. A typical light microscope has a resolution of about 200 nanometers, while an electron microscope can go down to 0.1 nanometers. That's a 2,000 times better resolution. Because of this, electron microscopes can magnify images much more without them becoming blurry or unclear. They're perfect for studying the tiny organelles inside cells, like mitochondria, which are way too small to see clearly with a light microscope. For example, an electron microscope image of a pancreas cell can show the nucleus right in the center, and mitochondria clearly arranged around it. So even though they're harder to use, electron microscopes let us explore a whole new level of detail, deep inside the cell. If this video helped you understand about microscopy better, make sure to like, subscribe, and hit that bell icon so you never miss a bite-sized science lesson from Edutube. Until next time, keep learning, stay curious, and remember, science is simple,